Well, thank you so much for that long introduction. 5 minutes feels like a long time when you're standing there waiting for your slot, but thank you so much for that. And what a delight and an honour it is to be here talking to you tonight. Now, my title is a long one, and when I looked at it, I thought I really could have done better. And so what I'm really going to talk to you about tonight is bringing innovation to infection control. Now, you don't often hear the word innovation in relation to infection control, but that's exactly what I'm passionate about and what I want to tell you about today. So what I want to do is to really set infection control in a much broader context. Now, I'm a clinician by training, a clinical microbiologist, and I'm completely blown away by the technology of the 21st century being used in medicine. And the first slide is, I wanted to really set the scene for that, to demonstrate where we are in medicine at a sort of pivotal point in where technology is really driving forward progress. So you may be aware of some of this, but some of the things I'd call out in medicine that are really revolutionary at the moment include robotics, machine learning, wearables, and virtual reality. So, for example, if you look at the slide of the robot, this is a picture of a robot that was taken at Cambridge Science Festival recently. And I don't know if you've come across these robots, but this is the Da Vinci robot, which can do a prostatectomy guided by a surgeon who actually sits with his head in this instrument, moving the arms of the instrument. This was developed not for human use, particularly patient use, but was developed for space and also for battlefields. But here we are with robots actually in most hospitals now. Now, if you go and have a chest X-ray or a head scan in the future, that is going to be interpreted by a machine. At the moment, it's interpreted by humans, but we are in the process, or people are in the process, of taking a large number of X-rays and getting machine learning to do better than we can. Now, if you're a diabetic, you'll know about wearables and what a huge revolution there's been in how you control diabetes. So you'll have a probe under your skin monitoring your blood sugar level, which is connected to your phone, which helps you to control your blood sugar. And those results can be fed back to your healthcare worker. And actually, I was blown away by this example, which I found only today. I was reading a blog by somebody called Roy Lilly, who blogs about the NHS. And he talks about fabulous things. And this is actually virtual reality used in Barrow Inverness. So this particular woman has a chronic pulmonary disease. And she, rather than go for physiotherapy at the hospital, she signed up to have a virtual reality treatment where she looks in these goggles and sees a beautiful Palm Beach. And she does her physio through the goggles, obviously moving her body, and that's all fed back to her healthcare workers. So these are just a kind of small snapshot of what is happening in hospitals and what you might get exposed to when you go into hospital next. Now, in that setting, infection control is a worthy target for innovation, but it's an innovation desert at the moment. Now, I want to say why infection control is important to you and me and your family. Now, this is one example of where infection control can really help patients. So this is a report from ECDC on surveillance of surgical site infections. So you go into hospital, you have your operation, and you've got between a 1% and 10% chance of getting a wound infection as a result of that, depending on your procedure. And that's bad for you or your family member. You're going to stay in hospital longer, your outcome's going to be poorer, you'll have a higher mortality. We also know that nosocomial infections, i.e. those that happen in hospital, they attack the most vulnerable patients, people in intensive care, people on cancer chemotherapy, and they're often associated with multi-drug resistant strains. And we know that when that happens from this WHO report, that if you're infected by a drug-resistant strain, your chance of dying is twice that of somebody infected with the drug-susceptible strain. So infection control is all about trying to stop those adverse events in patients in hospital. And a key plank of that is to really stop pathogen transmission. Now, you can be infected by a strain you've brought in with you, but often you acquire new strains in hospital, which can be selected out by you having antibiotics. And then they can cause nosocomial infection or a nosocomial outbreak. So infection control is really about trying to control onward transmission of bacteria. Now, I'd like to just demonstrate with 3 slides why I think that we're not doing that very well at the moment. And I'm going to go right back to the beginning of modern-day epidemiology, back to an outbreak in Soho in London in 1854, when John Snow, who's the father of modern epidemiology, was working as a public health doctor, and he recognized and he drew this map that a cholera outbreak was happening in Soho. So you can see here the map, you can see the blocks for people who developed cholera in that time. And then somebody has taken that information and actually plotted it on an epidemic curve with cholera deaths versus the timeline. And there's a couple of things I'd like you to see about this. First of all, this is a fairly typical epidemic curve. And in addition, when he took off that handle from the Broad Street pump, which was said to actually stop the outbreak, actually the curve was already coming down. And this says to me that intervention is often happening quite late, later than we would like. So this style of epidemiology is what we call shoe leather epidemiology. And actually these shoes are in a museum to demonstrate shoe leather epidemiology. He's obviously done a lot of walking in a CDC museum. This is how we do outbreak investigation. We go and look and gather information. Now, we're doing better now, of course, aren't we? Well, 150 years on or more, you may well remember just in 2011 the outbreak of E.coli that occurred in Germany. So this is an outbreak that the epicenter at the beginning was in Hamburg, and this is again an epidemic curve, shows the number of cases and the timeline. And what you'll see from this graph is that the dark blocks are the people with gastroenteritis, diarrhea from their infection. The lighter blocks are people that got something called hemolytic uremic syndrome. It's where your kidneys are affected by a toxin produced by the bacterium. And then the line is when notification started. And I'd like to sort of make the 2 points that I made earlier. One is that it starts quite late, and it really doesn't reflect, actually there's only really 1 point, and that is it starts quite late. Now, my ambition is to actually bring our attention to these really early cases and catch it before it really kicks off, and I think it's possible. But I've got one more case study coming right into the hospital now that shows that we're still not doing this in 2011 in hospitals. And this is not unusual. This is not because we're no good in Cambridge and can't do infection control. This is pretty typical. So in Cambridge, where I work, we have a special care baby unit. And we had a suspected outbreak. Now, just to describe this timeline, along here are days from 0 to 350, and every row is a baby. The length of the block is how long they were in, and then so you have patient 1 to 14. Now, on this unit, there were 3 cases of MRSA, P11, 12, and 13, which caught the attention of infection control. They thought there might have been an outbreak. So what they did as part of their investigation was to look back 6 months to say, have any other cases occurred? And this is when they revealed this cluster of patients. They took this very seriously, but they actually concluded there wasn't enough evidence of an outbreak. Now, I wonder what would have happened if we'd focused on those first 3 cases and actually got some more discriminatory information about them, and you'll see how we did that later. So what I'd like to say at this point is, our ability to really detect outbreaks is not very good, and it hasn't really changed in around 150 years. And what I'm going to tell you about now aims to try and change that. Now, I want to, with my colleagues, redesign hospital outbreak detection. That's the ambition. And so let's just think about what that entails at the moment. So you have 3 parts: surveillance, epidemiological investigation, and then if there's something to do, you intervene. Now, this is what really happens on the ward and in the hospital. So the first thing is infection control nurses. They get a list, a daily list of patients, everyone in the hospital with positive indicator organisms. What do I mean by that? That's methicillin-resistant Staph aureus or MRSA, Clostridium difficile, multi-drug resistant bacteria, things we don't want spreading to other people. So they have a daily list. And then they look for 2 or more cases that are positive for the same indicator organism, and then look for overlaps in time and place to see if a transmission event has occurred. Now, if you have 30 to 50 MRSA positive patients and you have patients moving around the hospital as fast as they do at the moment, you're hard-pressed to see the gaps or the overlaps between their contact. And I don't know if you've had any relatives in hospital recently, but with increasing pressure on accident emergency, very high bed use, patients are moving around the hospital very fast. So for an infection control nurse or doctor to see where the overlap is in time and place and go and fix whatever went wrong is a bit of a challenge. So the next thing they'll do, if they think something is kicking off, is to go to the ward and do an initial assessment, ask questions, get some information. And if they think there's something going on, call an infection control investigation. And at that point, they might consider bacterial typing. And they're unlikely to do it for two reasons. One, it's done in a reference centre. So it takes time to get the strain there and time to do the test. So it's about a week. So by the time you get the result back in a week, things have moved on. Two, bacterial typing at the moment is not very discriminatory. So let's just take MRSA. About 80% of all MRSA we isolate belongs to a single clone. We call it sequence type 22. You can't tell the difference between one and another. So most people have pretty much given up on bacterial typing in infection outbreak detection. At some point, some actions to contain further spread may be taken. What we are proposing is that we change all of that. We change the paradigm and that we do bacterial sequencing combined with epidemiological analysis. We remove the top section and we go straight into actions. Now, for bacterial typing, it's no longer in the relegated kind of to something that people rarely do. It's actually the first violin in the orchestra. It's actually doing the first thing you do in outbreak detection and investigation. And not only that, I'm going to show you in a minute that we can do this as fast, in such a fast period of time that it pretty much is the same time as doing this initial paper-based exercise. It's in a day. That's the revolution that we're talking about here. So I just want to give you kind of an overview before I go on to look at the evidence that underpins this. I've called this sequence first because we do the sequencing first, and that guides outbreak detection. So what we do is, in a microbiology laboratory, when a sample comes off the normal lab that's got one of these indicator organisms, say MRSA, we pick some colonies, and we extract the DNA, and we make a sequencing library. Now, you heard that we've developed some tests. We have developed this single colony whole genome sequencing test, which is there in JAC. So we then sequence. I'm not going to talk about the sequencing machines right now, but that takes a day, that whole pipeline. We then, on day 2, the morning of day 2 actually, we feed that sequence data together with the patient laboratory data and automate that to work out the patient movement and the genome sequencing information to see whether there's any overlap in time, space, and bacterial similarity. We're supported strongly by a collaboration with NextGen Diagnostics, who work at the Sanger in the Bioinnovation Centre, who've actually built this automated interpretation tool, which actually has allowed us to do this in the clinic. And we then have a sample report. It's MRSA, it's isolated or related to some other isolates from certain patients, and we can also predict resistance. Now, that's what the new technology looks like. And the laboratory report would be rather like this. We have patient details. We have a summary, so we know what the bug is. We know if there's a recent transmission. And then we cover aspects like organism susceptibility, and then the cluster detection. Now, this is really important because what we're doing is we're comparing similarity of one organism with another organism compared with the epidemiology. And this is where the magic comes in, because we're able to tell by comparing isolates whether the transmission likelihood is high, possible, or low. And this is completely understandable by infection control. So we don't need any informaticians sitting in the clinic. We hand this report to infection control. So what we're doing here is we're changing the paradigm in infection outbreak detection. And really, this is just to summarize why this is important. So we're doing active genomic surveillance of all the indicator pathogens, and it provides this very early warning of outbreaks that can be then contained. So it's a disruptive technology for infection control. So yes, we can innovate in infection control. And we call it sequence first or seek out. So what I'm going to do now is actually go back a step and give you the evidence for how we got to that point. So we've actually constructed this and it's ready to go. But I'm going to take you through some of the evidence to describe how we got on that journey. I'm going to look at the technologies that we've used, and I'm going to look at evidence that sequencing is better than current typing methods. I'm going to show you the first application of sequencing to an outbreak and then demonstrate that we can get clinical impact from doing that, because that's the whole point of doing it. And then first evidence that sequence first brings major benefit to public health. And finally, most important in an NHS context, the cost effectiveness of it. So a timeline for sequencing technologies. You may well be aware of this if you're using them yourself. But actually, around the time I was leaving medical school, the first automated DNA sequencer developed by Applied Biosciences was released to the market. And that was based on the technologies developed by Fred Sanger. By 1998, just over 10 years later, two academics in Cambridge were developing new technology, sequencing by synthesis. And they developed by 2006 something called a genome analyzer. They developed a company called Selecta. And Illumina actually acquired Selecta the next year. And the rest is really history. Illumina is one of the market leaders for sequencing instruments at the moment. By 2015, we know that Oxford Nanopore Technologies were releasing their instrument for commercial use, although there's been quite a long period of evaluation and improvement, particularly with the academic community. So the thing with sequencing technologies is that it really provides a tipping point for where we are in bacterial sequencing in relation to the clinic. So the cost has come down by a millionfold since its inception, and that's so critical for work in the NHS. And the speed is extraordinary, actually. So if we take a bacterial genome that's 4 million base pairs, in 2001, that took 18 months to sequence. In 2018, it will take less than a day. And we're routinely sequencing 24 genomes in 13 hours in a clinical setting, right in the microlab, using a Illumina MiSeq. And I'd also just highlight that Oxford Nanopore technology also gives you extra oomph in terms of long read and looking at plasmids. So what I would say is that really it is time for researchers to use this in the clinic. So I said that I would actually show you that sequencing is better than current typing methods, and I've got 3 quick case studies with a focus again on MRSA at the global, at the national and the local level. So if this study is absolutely groundbreaking. It doesn't feel like it anymore, but it was only 2010 when Simon Harris and colleagues sequenced 63 isolates. That's a drop in the ocean now, but that was really revolutionary at the time. He had 63 isolates of MRSA, a lineage that's the most important MRSA lineage across the world. We call it 239. And you can see the time of the isolates there. You can see the places where they were obtained. And you can see what the analysis was in effect. So the isolates were sequenced, and then they were sequenced with Illumina, so short pieces of short read DNA sequence generated, and they were compared to this reference genome, which was a perfect genome of an MRSA isolate. So all of the reads were kind of tiled onto that reference to allow single nucleotide polymorphisms to be picked up. This was just in the core genome, not the accessory genome that has all the genes that move around, the conserved core genome that's in all the isolates of MRSA or Staph aureus. So these SNPs, you gather these up to form a tree, and even a single SNP can distinguish between highly related isolates. So this is what Simon found. And I'd like to really pick up 3 things, really. One is these are all ST239 by multi-locker sequence typing, but there's huge diversity in the genome of these isolates. The second is something we call phylogeography. So if you look at the colours, you can see that isolates tend to cluster together by colour. And so this cluster is a South American cluster. This cluster is a sort of Asian cluster. So you get phylogeography on an international scale. The third thing I'd point out is that actually this technique showed us we can track intercontinental transmission. And we did that by these two isolates that appeared to come from Europe, but actually were in the Thai clade. And when we dug into these, there was very good evidence that these people had actually acquired their MRSA in Asia. So this was our first foray into sequencing of populations, bacterial populations. Does it work at the national level much lower? Well, we had a fantastic collaboration with BSAC with their bacteremia surveillance, and we got hold of 780 MRSA isolates. This time ST22, that's our UK lineage over 10 years. You'll see from the map the pins where we got the isolates from. So they're pretty broad. It's the UK plus the Republic of Ireland. And we sequenced them as before. We looked at the SNPs compared with the reference genome. And you can already tell that we can do a great many more isolates in one go or analysis than we could in Simon's paper. So this is a phylogenetic tree. It doesn't really tell you that much apart from the fact that it's quite an attractive one shape. But what we need to do is look at whether there's a phylogeographic signal. So we really need to map onto that some geography. So the way we did this was we used an idea that was originally suggested by Chibi Donker, where you divide up your geography by healthcare referral networks. And that means that people in, say, a particular block, they tend to get their treatment in that block of the country rather than go to another block of the country. So you kind of divide up the country by health networks. What we then did was we colour-coded those isolates in the head alone with where they came from. Now, ignore the circle around the outside and look on the inside and look for areas where the colour is the same. So what you'll see here is Wales, there's isolates there. You can see Ireland and Northern Ireland, a big block there. And if we then map that back to the map, what we'll see is fantastic phylogeography. So what that tells me is I can tell where an isolate has come from, which in transmission tracking nationally is really important. So yes, we can see these changes nationally, but you might actually say, well, this is over a decade or longer. Of course you're going to get changes in the bacteria. What we really want to know is the ward level. So this is what this study does. We based ourselves on 2 wards in intensive care units, actually, both of them in a hospital in Thailand where I was working at the time, 1,000-bedded hospital. And we swabbed everyone who came in, and then we swabbed them twice weekly and the staff. Now, we didn't know what MRSA carriage rate we were going to find here. It's the first time we'd ever looked. But we recruited, you can see the number of people we recruited here, and 1/3 of children were carrying MRSA, and 16% of adults were carrying MRSA, and staff were carrying MRSA, and they were all ST239. So we sequenced isolates, and we added another 20 isolates from the last couple of years from that hospital. And so this is what we found. Now, I'd like you just to look at this, if I can get my pointer to work, yeah, this block here to start with. Now, what you'll see here is that each isolate is labelled with the ward that it came from. So you can start to see clusters of red wards, that's the adult, and blue, that's the paediatric. So even on the ward, we can tell which ward effectively that isolate has come from. You'll see that the staff are actually scattered throughout that, which is kind of what you'd expect. And the bloodstream isolates, which are a bit older, are, as you'd expect, ancestral to these more recent lineages. The second thing I'd like to say is that we were able to kind of classify most of the isolates into these 5 clades which are colour-coded. Now, the reason we wanted to colour-code them was we wanted to actually look at not only the clade in a given point in time, but over time, which is what this next slide does. So if you focus on this particular figure here, what you'll see is a timeline from 0 to 90 days. That's the duration of the project. And down here, along here, you'll see the patient numbers. So the first patient that came in was T9, and they were positive for MRSA in the blue lineage. And then the second patient acquired that blue lineage. Then the third patient came in with the pink lineage, and on and on and on throughout the study. So you'll see that at the beginning there was quite a lot of diversity, but by the time we got to the end of the study, this patient appeared to be feeding this entire carriage situation, or at least was the initial seeder for these particular patients. Here, it's the adult ICU rather than the pediatric ICU, and you can see the same sort of thing. So you can see this really dynamic influx of different lineages and throughout the study period. But I think these are kind of the more intuitive figures. What you can see here is that rather than just saying these are all ST239, the two wards look completely different, and you can see actually over time the number of MRSA positive patients with these different lineages. So extraordinary resolution for surveillance. So I was happy after having done those 3 studies with my colleagues and team that this technology was far superior to current methods. So we then applied it to an outbreak. This was the first application of sequencing to an outbreak reported, and it was a really simple study. We got 7 people who were involved in an outbreak, 4 MRSA isolates from people that were not involved in the outbreak, and sequenced them. And you'll see from the tree that the outbreak and the non-outbreak isolates are really separated. These are the outbreak isolates there. So we were confident we could actually split isolates apart. This is what we predicted, but we hadn't actually done it for real. We also found that we could predict phenotypic antibiotic resistance. So this is a cluster of genes all put together here. These are drug resistance encoding genes. These are toxin genes. And we searched the genome for these genes and actually found that, of course, they're all MEK-A positive, which encodes methicillin resistance, which is why they're MRSA. So the blue is negative, the red is positive. And this was actually the first demonstration that you can work out if an isolate is resistant or susceptible to an antibiotic from the genome. So we step up again. What we want to do is show clinical impact from sequencing. We can do it, but actually, does it make a difference to the patient, which is the critical thing? So this is the next key study that we reported in the Lancet Infectious Diseases, where we went back to that outbreak in Special Care Baby Unit to explore much further. So if you remember, this is the figure that I showed you initially with these 3 cases where they were a concern to infection control, but this was not considered to be an outbreak by infection control. The first thing we did was sequence them. Now, this is a slightly different tree. If you look at the legend here, all this is SNPs from the root. But what I can tell you is that these isolates are really very related. So the first thing I can say is that this is an outbreak. Categorically, we can say that. Now, at that point, we were interested in seeing about transmission, whether this extended beyond the Special Care Baby Unit. And so I won't go into detail, but we found more isolates that we thought belonged to that lineage, and we sequenced them. And looking at the phylogenetic tree, all 10 isolates fitted in with this outbreak. Now, at that point, we knew nothing about the clinical provenance of those isolates, where they come from. But what we knew on the timeline was that actually they went across the timeline here. And you can see from the symbols that they weren't all babies. Some were adult men and women. So we looked in their notes. We went to their notes to find out how they linked up. And this is what we found. So not only did we have an outbreak in SCBU that we didn't know we had outbreak into the community, which we didn't know we had. So I just want to walk you through this. So this is the tree, which is kind of color-coded for these lines. So here, you'll see the original SCBU cases. These are people who were in SCBU, but we thought they were MRSA negative because we screen people every week. We never picked it up. These are women in the maternity ward who were MRSA negative when they came in, clearly not when they went out. And this is people in the community. And by comparing their medical notes and their phylogenetics on the bacteria, we could actually link cases together. So let's just take this baby here. So this baby, this part of the tree, this baby is likely to have passed on MRSA to mum and also to dad. In this instance, this baby, who we didn't know was carrying MRSA, passed this to mum, who passed it on to another mum, who passed it on to their partner. what we can tell from this is that we have incredible forensic power to see what is spreading where. Now, there was one patient who was kind of an outlier. We didn't know how this linked up, but then something happened. Another baby came into special care who was MRSA positive. Now, this is really odd because there's a very long gap between this group and this new baby. It was over 60 days. And so we thought this has to be a healthcare worker. So to cut a long story short, we swabbed 160 healthcare workers that were in and out of SCBU working with the babies. And we found somebody who was MRSA positive. We sequenced multiple colonies from their nasal swab, and here they are on the phylogenetic tree surrounding this baby. The impact here is that we removed that healthcare worker from their workplace whilst we decolonized them from MRSA, after which they went back. And this outbreak simply stopped. Now, I'd like to look at consequences of this outbreak. Now, they were quite severe. There were several women who actually had very severe breast abscesses who were breastfeeding. This is not a time when you want to have a breast abscess. And one woman had such severe disease, she had to go to theatre to have her breast debrided. The number of times they went to emergency department, GP visits, etc., was extensive. And what I would say is that rather than saying there might have been an outbreak here, why can't we say there was an outbreak there? That's what we're working towards. Now, just out of interest, I don't know how many of you have young teenagers in your family who are doing A-Level Biology at the moment, but this has worked its way into the OCR textbook. I didn't know actually till Julian sent me this link, but they've actually picked up on this as a case history for people doing A-Level to learn about. And so I'm pleased to see that, that your children will be learning about bacterial genomics. So, moving on to evidence for sequence first. We want to sequence the isolates as they're cultured in the laboratory, but what's the evidence for this? Well, the evidence comes from a very nice study reported in Science Translational Medicine. Francesc Cole, who's in the audience, did a lot of this work, and he did a fantastic job on it. So it's quite simple. We stayed in a laboratory for 12 months, a big diagnostic microbiology laboratory for 12 months at Addenbrooke's Hospital, which served 3 hospitals and 75 GPs. And we identified everybody who was MRSA positive over the course of a year in 2012, 2013. And then we sequenced at least one isolate from every case. And we collected their epidemiological information. Was a sample sent from a GP? What's their postcode? Have they been in hospital? What wards have they been on? And so on. So this is recruitment statistics. And you'll see here that we did this for a year. We recruited 1,465 people. We sequenced more than 2,000 isolates from them. And you can see that most people came from one of the big hospitals, but we did have quite a few patients where samples had been sent in by their GPs rather than from hospital. Most people had no disease, in that this was just a screen. It was just to see if they were positive or not. Some people had clinical disease and some people had both. And this simply shows you the distribution of our cases with a heat map for how many cases of MRSA were from each part of the region. Now, I'm not going to go through this. I really want to use it to describe how complicated the integrated data analyses were at that time. This took 4 years to analyse and to get into print. But you'll see there's 4 compartments in this. There's the microbiology, there's the genomic analysis, where we're doing all the sequencing and doing all the informatics. There's the epidemiological analysis, collecting all the patient movement, and then trying to bring it all together to see where people have overlapped and whether they've got isolates that are similar to each other. So this is very, very time-consuming if you're doing it in this way. Just to zoom in on the genomic and epidemiological integration for a moment, what you'll see here at the top is an example where 2 isolates are very closely related in a phylogenetic tree. And in each case, Francesc looked at their epidemiological link. And for example, in this case, these 2 people lived in the same house. In here, where the isolates were very related, he noted that this particular cluster had occurred through hospital transmission. So by this, building up slowly over time, we knew how those 1,465 patients were or were not related. So here comes the punchline, how many people were related. So this is a phylogenetic tree of just the ST22, which is 3/4 of the collection, and it's color-coded by whether there was hospital transmission, community transmission, both, or we couldn't detect any. And I think the key thing is what comes up on the screen now. Now, we found that 40% of all the cases were linked to 173 different outbreaks, none of which were known by infection control. So we found 173 new outbreaks, which involved between 2 and 44 people. Some were in hospital, some were in the community alone, and some were people, it included clusters with people from both environments. So this is a staggering amount of outbreak detection, which is going undetected. Now, I said that some transmission was going on in the community, and I want to pick up on one particular outbreak, which involved 27 people. You'll see the arrow there. They were from the same GP. They had no links to hospitals, and so we went and investigated that as a separate investigation, and that work was done by Michel Tolman and published in Clinical Infectious Diseases. Now, this goes back to shoe leather epidemiology and John Snow, in that we collected what clinics they'd been to at what time, and we looked at 4 different clinics. We looked at and GP visits. We looked at specialty clinics, the community podiatry, and so on. And what we found actually was that this all focused on podiatry and diabetes ulcers. So these people had acquired the MRSA most likely during that period of time. And unfortunately, one of those patients developed bacteremia and died. So what do we learn from Sequence First? Well, infection control practice in hospitals is not sufficient to prevent MRSA transmission. We're getting MRSA transmission in the community, which is being introduced into hospitals. Infection control in GP surgeries is not sufficient. And really, community acquisition could have been detected by integrated data in the diagnostic lab. So moving towards the end of my talk, this is all well and good, but how are we going to afford it in a really cash-strapped NHS? So we've done an early model of cost effectiveness where we've looked at standard of care, which is you can't do anything about the outbreak cases 1 and 2. That's the first case you detected. But if you have standard of care, based on the previous study I've shown you, you'll likely get transmission to cases 3, 4, and 5, and so on. A proportion of those will go on to develop MRSA infection, and this is where the cost comes in. So there's no real cost before that, apart from cost to the patient. But when they develop an MRSA infection, that's expensive to the patient in terms of time and their outcomes. It's expensive to the hospital. So that's standard of care. If we look at Sequence First, what we want to do is get the first 2 cases and go in and clean up. So we won't get transmission to cases 3, 4, and 5, and we won't get MRSA infection, and this is where the cost saving comes. So we have got an early model. This is from the literature evidence and Francesc Coll's work. That's the input data, that's what goes into the data. We've got a decision tree model to estimate the impact. And the key thing is we're looking at relative reduction of MRSA acquisitions and infections by Sequence First. It's a pity I can't ask you how much you think you're going to save from doing this, but it probably won't work in this venue. So the upside for every patient admitted to hospital is £18. And for 60,000 admissions every year, which is what we have in Addenbrooke's, that's 1.1 million of savings. So the ratio of cost versus cost benefit of Sequence First is about 1 to 3. It's true that you have to spend some money up front to get the equipment in, but this is, at least in the early model, very convincing that NHSs can afford this, and it's good for their patients. So, next steps. We are going to move from the John Snow shoe leather epidemiology, and we're going to move on from this weeks, months, and years of analysis where we take 4 years to do a study, and we're going to be doing this. We're going to be sequencing MRSA in 24 hours in our laboratory and feeding the results back to infection control. We've got a study coming up shortly. We're going to try this for 3 months in the first instance. We're going to sequence every day, Monday to Friday, routine. And then we're going to say, how related is the test isolate I have here to all the other isolates I've sequenced in the last 3 months? We're going to take the daily results to infection control, and then we're going to track what they do as a response, and then we're going to model cost effectiveness and define impact. So what I'd like to say in close is that this is innovation coming to infection control. I believe it's going to make a big difference to how we manage patients with MRSA carriage and infection. I'm really excited about the following 6 months when we're doing this study. This is perhaps the most important slide, the acknowledgement slide. So I would thank, from the bottom of my heart actually, the Peacock Group. Francesca Coll is in the audience, as is Catherine Ludden. But you can see people, Beth Blaine and Cathy Raven, particularly in the laboratory. Public Health England, in our laboratory, we were really critically involved with working with them to get the material from their laboratory. BSAC gave us isolates, and Wellcome and Simon Harris were really critical for some of the sequencing. We're working with York Health Economic Consortium on the modelling. And I'd like to acknowledge my funders at the bottom here, but in particular, I would like to acknowledge the importance of our commercial collaboration with Next Gen Diagnostics, but two very special people who've helped me a lot in the last 10 years, Gordon Dugan and Julian Parkhill, who's also in the audience. So thank you.